Hello friends, again welcome to my channel. Now today we are discussing about one of the important component which is present within the all cells, that is either these are the eukaryotic cell or the prokaryotic cell, that is the ribosome. So today we are going to discuss about the structure of ribosome and their specific functions within the cell. So let us start. Now, whenever we are discussing about these ribosomes, now these ribosomes, these are made up of protein and the ribonucleic acid, that is the RNA. Means simply these are the ribonucleoproteins, that is the ribonucleic acid along with the protein molecule. These are considered to be the ribosomes. Means the amount of the ribonucleic acid and the protein, it is same within the ribosomes. These ribosomes, these are considered to be the tiny spheroidal dense particle which has the diameter of 150 to 200 angstrom. They are primarily found within all prokaryotic and the eukaryotic cells. A ribosome, it is made from the complex of ribonucleic acid and the proteins, and therefore the name it is the ribonucleoprotein, or simply they are called as the ribosomes. Around 37 to the 62% of the RNA, it is comprised of RNA and the rest it is the protein. Means within the ribosomes there is the presence of the 62% of the RNA, 37 to 62% of the RNA, and remaining part it is made up of the protein. Now majorly these ribosomes, these are get divided into the two subunits. Means these are made up of the two subunits, that is a smaller subunit and one it is the larger subunit. On the basis of their size, majorly these are get divided or these are get named as a smaller subunit and the larger subunit. Now this smaller subunit, it can binds to the larger subunit and the mRNA, and the larger subunit it binds to the tRNA and the amino acid, and with the smaller ribosomal subunit. Majorly these ribosomes, as we know that these ribosomes plays an important role during the translation process. So within that they are including the binding with or their interaction with the transfer RNA, then the mRNA molecule, these are involved. Now within the prokaryotes there is the presence of 70S ribosomal subunit which is responsible to carry out the process of translation, which is made up of the two different subunit, that is the smaller subunit it is considered to be the 30S ribosomal subunit, and the larger subunit it is considered to be the 50S ribosomal subunit. Now within this prokaryotic ribosomal subunit, it is made up of the 16S ribosomal RNA. This 30S ribosomal subunit, it is made up of the 16S ribosomal subunit, that is approximately there is the presence of 1540 nucleotides, and to that there is the involvement of 21 different protein. There is involvement of the 21 different protein. Now in reverse to that, whenever we are focusing on the another component of the 70S ribosomal subunit, that is the larger subunit, that is 50S ribosomal subunit. Now this 50S ribosomal subunit, it is composed of a 5S ribosomal RNA, which is made up of 120 nucleotides, then the 23S ribosomal RNA which contains up to the 2900 nucleotides, and the 31 different proteins. 31 different proteins. Now both of these two subunits, that is the 30S and 50S subunits, these are fit together and majorly these are work together during the translation process, that is they are responsible for translating the messenger RNA, and there is the synthesis of polypeptide chain. Majorly both of these subunits or both these complete ribosomal subunit, that is the 70S ribosomal subunit, it is these are formed from two different subunits which are non-equal in size, that is the 50S and the 30S ribosomal subunit, these are non-equal in size, and the 50S ribosomal subunit, it is larger in size. So due to that, majorly whenever there is the formation of this complete ribosomal subunit, they are slightly longer in the axis than in the diameter. During the protein synthesis procedure, multiple ribosomes, these are attached to the same mRNA molecule. There is attachment of multiple ribosomes to the same mRNA strand, and this structure it is called as a polysomes. Means whenever we are observing a structure that is a linear mRNA chain to which there is the attachment of a multiple ribosomal subunit, then it is called as the polysome. The existence of these ribosomes it is temporary, that is only for during the process of translation. These subunits, these are joints with each other, that is 30S and 50S subunit, these are joints with each other and forming the 70S ribosomal subunit. After the completion of translation process, there is the separation of this subunit, and majorly again they are reused for the next cycle or next translation cycle. Now in reverse to this, majorly in case of the eukaryotes, instead of presence of 70S ribosomal subunit, there is the presence of 80S ribosomal subunit, and within that the smaller subunit it is made up of the 40S ribosomal subunit, and the larger subunit it is of 60S ribosomal subunit, and combinedly these are responsible for forming the 80S ribosomal subunit. Now here it is the structure of prokaryotic ribosomal subunit, which contains both the small and the large ribosomal subunit. Now within that you can observe that within the small ribosomal subunit there is the presence of the three different sites, that is the E site, P site and the A site. And majorly the smaller ribosomal subunit it is having a specific sequence through which they can joins with the small ribosomal, they can joins with the messenger RNA. That is, on the messenger RNA there is the presence of a translation initiation sequence which is called as the SD sequence or the Shine-Dalgarno sequence, and its complementary sequence it is present within the 16S ribosomal RNA, which is the component of 30S ribosomal subunit, and due to that this 30S ribosomal subunit it can binds with the messenger RNA. Along with that, within this 30S ribosomal subunit, these three sites these are present, that is A, P and E site. To the A site, majorly it is the site for the attachment of incoming amino acylated tRNA molecule. Amino acylated tRNA molecule. At the P site, there is the site where there is the formation of peptide bond in between the two amino acid. Here in this figure you can observe there is the formation of peptide bond in between the tryptophan and the methionine. And at the E site it is the site from which the inactivated form of the tRNA or the deacylated form of the tRNA molecule, means whenever it transfers its amino acid to the P site, then it is get removed, this deacylated tRNA molecule it is get removed from the E site. So here it is the function of this small ribosomal subunit. The large ribosomal subunit, this large ribosomal subunit has the peptidyl transferase activity. That means the formation of peptide bond in between the two amino acid for the polypeptide chain synthesis, it is get stimulated by the activity of this large ribosomal subunit, that is the 50S ribosomal subunit. Now we are switch over to the what are the specific functions of these ribosomes. The ribosomes, these are considered to be a complex molecular machines which are found within the all living cells, either the prokaryotic or the eukaryotic cells, and majorly these are acts as an sites where there is the process of protein synthesis can occur, or these are responsible for providing the platform on which the protein it can be synthesized within the prokaryotic and eukaryotic cells. These ribosomes, these links the amino acid together in the order specified by the messenger RNA. Means on the basis of sequence of the codons which are present on the mRNA molecule, there is the transfer of the amino acid within the growing polypeptide chain. These ribosomes, this can be acts as an catalyst in two extremely important biological processes, that is called as the peptidyl transfer and the peptidyl hydrolysis. Means for the purpose of formation of peptide bonds in between the two amino acid, which is catalyzed by the 50S ribosomal subunit. So here it is the somewhat information about the ribosome, their structure and function. So if you like this video, please subscribe my channel and also share this video with your friends. Thank you.